We're going to start today with a presentation by Ryoya Nishida. He's the co-founder and CTO of Towing, a biotechnology startup from Nagoya University, Japan, specializing in microbial technology. He's going to be presenting on his work developing microbe-immobilized biochar to boost the use of organic nitrogen and stabilize soil pH. Ryoya?
Yeah, thank you for the introduction. Hello everyone, my name is Ryoya Nishida, founder and CTO of Towing. So this is my second time presenting at this conference. Last year was really exciting. I'm looking forward to connecting with many of you again this year. So the title of my talk is Improving Soil Health Through Enhanced Organic Amendment Use and pH Regulation via Microbe-Immobilized Biochar. So first of all, let me introduce our company. We are a soil microbiome technology company spun out of Nagoya University, located in Japan, developing biochar-based biological soil improvement technology with a mission to create circular and sustainable agriculture practice. And our team includes 4 groups: agronomy, soil science, biology, and bioinformatics. And the Regenerative Agriculture Lab as industry academia collaborative chair in Nagoya University. Together we develop microbe-immobilized biochar. Let's move to today's agenda. Firstly, I will introduce the trend of biochar with microbes and our key technology and R&D activity, and our product Sorotan as well. Next, I'd like to talk about the key topic in soil health in relation to Sorotan. So let's start with the biochar effect. So as many of you already know, the use of biochar in agriculture has many positive effects. It promotes plant growth, markedly improves soil properties, GHG reduction, and heavy metal remediation. However, several studies have also reported negative impacts, such as excessive increase in soil pH and negative changes in microbial community related to the carbon and nitrogen cycles. So there are a lot of factors about this, but they depend strongly on pyrolysis condition and feedstock types. Biochar may have a strong liming effect and contain pollutants like VOC and PAH and so on. So this is a key theme of my presentation. Can microbe mitigate negative and boost positive effect of biochar? So biochar with microbes has been gaining attention nowadays. The use of biochar as a carrier for microbial inoculant has been shown to promote the persistence, survival, and establishment of the inoculated microorganism in soil and at plant root by providing a shelter-like habitat. So as you can see this figure, direct inoculation into soil is red square, and inoculated biochar is blue one. So there are large difference in surviving microbial population. But still there are a lot of challenges and limitations. For example, we need large-scale field experiment, developing cost-effective technique for large-scale microbial immobilization, and we have to adapt to unique properties of soil and biochar. And when it comes to sustainable agriculture, evaluation of soil nitrogen and carbon metabolism are largely lacking. So to address these challenges and limitations, we are building a commercializing platform for microbe-immobilized biochar. It includes methods to create a microbe-immobilized biochar using specific strain or microbial consortia, and application method to activate and improve target soil function as needed via ecological transplantation or ecological enhancement. So this image is inside of one of microbe-immobilized biochar. This clearly shows how biofilm form inside the pore of biochar. We control like this microbial colonization on biochar and in soil. Oh, sorry. Yeah, so in this platform, we are developing technology of microbial-immobilized biochar suitable for local materials, functioning for organic conversion and disease suppression and yield increase and reduction of environmental impact. So there are two main activities. One is microbial cultivation database for biochar. By controlling pyrolysis structure, acid, and organics, we aim to identify core microbes responsible for the key function of microbe-immobilized biochar. So we already conducted over 400 types of biochar and clarified the relationship between biochar properties and microbial growth. Another is biochar and microbe-immobilized biochar soil effect database. We are aiming to quantify the effect of microbe-immobilized biochar when used as a seedling soil or soil amendment, and to clarify the correlation between its function and various impacts through multiomics analysis. So this effort led to Soratan, one of our products. We are making Soratan by using locals, aiming to improve soil health, replace chemical resources, and reduce GHG emissions. So rather than combining a few selected strains, we successfully established a diverse and complex microbial community within the biochar matrix. Yeah, next is Soratan's main features. Soratan's microbial community consists of over 1,000 species with ammonifying and nitrifying microbes. So these microbes can help improve nitrogen use efficiency of organic nitrogen and suppress soil-borne disease and improve microbial community robustness. Yeah, that's because different microbes become active depending on the soil conditions, thanks to the diversity of the microbial community. Yeah, so using Soratan, we provide two main values, improving soil rapidly while accelerating pathway of biochar-based carbon removal. Yeah, so from here, I will present case study in Japan and in US. Yeah, so let's start with basic pot trial in Japan. Here are the effects after application into the soil. This is just an example. We use two types of soil, andosol and red-yellow soil. So we evaluated nitrate production and pH changes during incubation after applying Soratan or biochar and organic fertilizer. So in nitrate production graph, from left to right, soil only with biochar and with Soratan. And in pH graph, blue line is biochar and orange one is Soratan. As a result, with Soratan, enhanced nitrate production and from organic fertilizer and suppression of excessive pH increase were confirmed, regardless of soil fertilities. So this may enhance the utilization efficiency of organics and allow for higher application rate of biochar. So in addition, we conducted over 600 trials using Soratan in commercial field in Japan. Among these, showing an average yield increase of over 20%, even when replacing chemical fertilizer with organic input. Furthermore, the following insights were obtained regarding crop type and fertilizer, limited yield improvement in cabbage and soybean due to reliance on chemical fertilizers. And regarding soil condition, similar to biochar, the effectiveness is reduced in medium texture soil. However, pH appears to have a minimal impact. So to maximize the effect, we are currently developing optimal application guidelines for different crop and soil type. Yeah, so let's move to US project. Last year, we successfully replicated Soratan in the US and conducted a tomato trial in California via CRO. We tested 3 treatments, chemical fertilizer as a control, and almond shell biochar, and Soratan. And at this time, we caught a severe heat wave, and it reduced tomato fruit set. Thus, as shown in this yield result, the control yield decreased below the usual level, while the biochar treatment increased yield markedly, and Soratan showed better growth and an even higher yield than biochar. So we explored potential drivers based on soil physical and chemical property and the microbial communities. Yeah, so here is the result of soil physical and chemical property after harvesting. We measured basic items such as saturation percentage, pH, EC, CEC. So in this graph, from left to right, control, biochar, and Soratan. And compared with biochar, Soratan suppressed the rise in pH and EC. And like biochar, it increased SOM, water retention, and nutrient holding capacity. Yeah, so next we show post-harvest soil microbiome result. At the film level, taxonomy profile looks no large difference. However, regarding bacterial diversity, it decreased with normal biochar, but increased with Soratan. So we then analyzed in detail how Soratan shifted community structure and which function likely changed. Yeah, so this shows film level taxa with significant difference among treatment identified by ANCOM BC. Here, increase with Soratan are blue and decrease are red. By using this analysis, we found there are large difference between treatment. So Soratan significantly enriched plankton mycetota and chlorophlexi and bacteroidota and Pateski bacteria and Disulfobacteriota and NB1J, and reshaped the community composition toward groups linked to nutrient cycling and organic matter turnover. And biochar also increased some taxa with this function, but it tends to favor more oligotrophic, stress-tolerant taxa. So next, we used principal coordinate analysis to assess bacterial community shift. Here, Soratan is green, and biochar is red, and control is yellow. So Soratan separated from control and biochar. And in this analysis, arrow indicated, so in this analysis, arrow indicate key microbe driving the community shift. So Soratan's microbial community was affected by plankton mycetota, Firmicutes, and Actinobacteriota, and Gemmatimmonadota. So this shift raised diversity and function for CN cycling and plant growth promotion, indicating higher soil fertility and resilience. Yeah, so next we use biomarkers to predict in detail how these community shifts change soil function. We analyze three main functions: nitrogen cycle, and carbon degradation, and phosphorus metabolism. Yeah, so using taxonomy-based biomarkers, Soratan show markedly higher potential for all of these functions than control and biochar. Yeah, so next we show a heat map of pathway in relation to soil health by using P+2. Consists with the community shift, Soratan tend to increase nitrification and carbon degradation and carbon metabolism and phosphorus metabolism. And while reducing denitrification. Overall, Soratan strength, soil health function versus biochar and control. So in addition, we analyze the possible performance for drought and heat tolerance. We use Sankey diagram to visualize predict enzyme function for drought and heat stress tolerance. So in this figure, this showing treatment to enzyme to function flow are shown, and thicker flow indicate higher function potential. So Soratan channel more flow into ABA, GA biosynthesis and osprotectant production and polyphosphorus biosynthesis and loss detoxification and indicating improved heat and drought drought stress adaptation capacity. Yeah, so here, field trial summary, we attribute the substantial increase in the suratan treatment to the following factors. So regarding physical and chemical properties, biochar-like improvements in SOM, water holding capacity, and nutrient holding capacity, and mitigation of biochar-induced excessive increase in pH and EC. And regarding biological properties, mitigated the biochar-induced decline in bacterial alpha diversity and further increased alpha diversity. And large microbial community shift suggests enhanced function in nitrogen cycling and carbon metabolism and phosphorus metabolism and PGP activities. And community reasonably indicate improved tolerance to heat and drought stress, suggesting greater stress tolerance. Yeah, so before closing, let me briefly introduce our other countries' trial and future works. We have also successfully produced suratan in Brazil, Thailand, and Mexico. We are currently starting field trial in each country to build sustainable supply chain with food and beverage companies. Yeah, so especially in Mexico, we got preliminary results. It has shown increased SOM and yield than biochar in head lettuce while mitigating high pH and also high salinity. So we are evaluating improvement in soil microbial function and investigate whether suratan can contribute to soil health in each country. Yeah, so as future work, we are working on the following project with focus on regenerative agriculture and enhancement of specific function. So especially we are updating microbe-immobilized biochar for soybean. So these images are preliminary result. So the immobilized biochar treatment shows more nodules than direct inoculation. Yeah, and for mitigation of continuous cropping problems and boosting carbon fixation, like in combination with ERW and suratan and co-compost. Yeah, so here is the summary of my presentation. So microbe-immobilized biochar is a key enabler for soil health and sustainable agriculture. And what microbe-immobilized biochar enables is improving microbial establishment and persistence in soil and boosts the positive effect of biochar and help buffer potential negative effects. And regarding suratan, it can improve soil physical and chemical properties as with conventional biochar and increase the utilization efficiency of organic nitrogen from organic fertilizer and compost and moderate excessive pH increases and restructure soil and enhance microbial diversity and adds strengths soil function such as nutrient cycling and plant growth promoting and resilience. Yeah, so microbe-immobilized biochar offers a scalable pathway to more resilience and productive soil. Yeah, so thank you for your attention. Yeah. Application rate? Okay, yeah, thank you for your
and methods. Sorry.
Okay, yeah, thank you
Are
for
you
your
broadcasting or are you bearing and tilling in? Yeah.
Sorry, could you say that again? Last phrase?
I'm just curious about your application methods and rates.
Mm-hmm. Okay, yeah, so we are using suratan, just spreading and tillage so similar to normal biochar, and application rate is depending on crop type and soil type, but usually we apply suratan around 4 cubic yards per acre. Yeah, but we usually combine suratan with biochar, so yeah, regarding biochar application rate will be like around 5 ton per hectare. Yeah.
So, sorry.
Spreading, yeah, and tilling. But it depends on crop type. And regarding preliminary crop, we usually do just spreading to surface area. Yeah, yeah.
So I have two questions. I apologize, I came in late, and so maybe you covered this at the beginning of the talk. So you inoculated this biochar with these different strains of microbes, and then in the end you were testing for different microbial communities. Did you track those amplicon sequence variants from, so the microbes at the end, the ones that you inoculated with, did you track the microbes in the sorotan to see if those were the ones providing this additional functionality that you observed?
Okay, yeah, so that's very good question. And it's a bit difficult to track, so the microbe is fixed this soil because we usually use natural microbes, so it can be bit difficult to separate it from our product or it's from nature. But when we use specific microbe, we can track this survivalness.
Right.
Yeah, but in our research, we are using NZS and qPCR to track our, like our product can promote microbial activities and also microbial diversity. So yeah.
So, and then you were talking about, you were using, I assume, metagenomics to look at all these different enzymes.
Yeah.
So, and then predicting kind of what those enzymes would do. Did you observe that, those phenotypes you observed by looking at the enzymes, did you actually observe those functions in the soil, like improved carbon cycling, improved phosphorus solubilization, those sorts of things?
So you're asking about these.
So if you look,
function?
no, go forward.
Forward, okay.
Yeah, one more.
More.
So here,
Yeah.
so you're saying that you saw, ABA biosynthesis and polyphosphate biosynthesis. Did you actually see those phenotypes develop in the plants? So did you look at the plants to see whether or not they had increased proline or anything like that to sort of validate the shifts in enzyme production?
Yeah, actually, so we are doing this trial in our laboratory, so it's a bit difficult to do that in field trial. So but in our laboratory we can check this activity directly. So so similar, so how can I say, similar activity already observed in Japanese laboratory trial. Yeah.
Very cool. Thank you so much. I loved your presentation.
Yeah. Thanks so much.
I have one question. When you're using, at least from my viewpoint, it's pretty high rate in the field. My question is how much it costs. When you inoculate this microbiology, I mean the microbial into biochar, is it very costly or?
Thank you for your question. So, so it's very depending on biochar's cost. Now in US, we aim around $250 per ton. But for now, it's a bit difficult to set this price. So I hope biochar price will be not so expensive.
I just wondering, when you inoculate this microbial or bacterial into the biochar, is that very costly processing or just very simply?
It's not so costly process. So because we used immobilizing process like compost. So it's very similar process to composting. So it's very not so expensive process. Yeah.
Okay, thank you.
Yeah, thanks so much.
Does it have to be applied every year, or is it once and done?
Oh, sorry.
you have to apply for soil plan every year, or is it one?
Yeah, so our standard application, we apply once per year. Yeah, but regarding just using Soratan, it's around 1 ton per hectare, so it's not so expensive.
I have one question. Very nice presentation. I saw in some parts that the pH, you can increase the pH. In some cases, you can lower the pH, and it's good for salinity soils.
Yeah.
So, what do you think the effect is in which case? Because they are like opposite in some cases.
Yeah, so thank you for the question. So regarding pH, yeah, when we apply our product in like high pH soil, so the ratio of Soratan, so we apply high ratio of Soratan, and when we use our product in salinity soil, we usually combine Soratan with biochar to adapt pH level.
Sorry, can you repeat? You have high salinity, you apply
So high concentration of Soratan.
of
Soratan. So yeah, so Soratan is mild biochar because so they have buffer capacity and they have microbes. So when we apply Soratan for high salinity soil, it can be work like mitigate this high salinity and so mitigate pH increases. Yeah, so I'm in our booth in first floor. So yeah, if you have any questions, so please feel free to ask me and let's discuss about this Microbe-Immobilized biochar. Yeah, thank you so much.